And, oopsie daisy. And we're going to have a test on Thursday. And we do have some more new material that will be on our test from Chapter 19 that we'll talk about today. And we'll have time to do that. And we'll have time to talk about all the questions you have as well. Yep, and so let me just double check this situation.
Do you
All right.
have your triangle recorder thingy today?
Nope, sorry.
So should we record the class?
Nope, it's, that is an external microphone that does a better job picking up student questions, you know? And I just didn't have it in my backpack today, and, but I have my internal microphone, and that does an okay job.
Thank
Yep.
you.
Yeah, okay, and so, so we have time to answer questions. Let's just start out by saying we have our exam on Thursday. Sarah asked me, will I send out another practice exam? No. That first exam, I always send a first exam out for the first exam on a course so students can see what is going to be expected. But you know now what our second exam is going to look like. It's going to be 50 questions, very much same style format as the first exam. And that's it, right? So that's where we're at on that. That's what the second is going to look like. We'll want to be mindful of our time and, work through our answers, right? And pay attention that, each one's 2 points. Then I made another little video to help with the term paper. You're probably not working on it right now because you're studying for the exam, but we got to roll ahead on that. So I'll get that posted on Brightspace too, because I've had some specific answers that I think are specific questions I think are general questions about, well, what are the details of the term paper? And so I'll get that up on Brightspace today. And so that's where we're at. And we do have this new material to talk about. Oh, and so students have submitted this application, and that's great. The deadline is passed, but if you're interested in that anytime, just let me know. Year round, I'm trying to help recruit students. So we can jump into this, but a couple of students are asking questions before the class started, and I'm sure there are more questions. And so why don't we see about questions that people have, and we'll go with that for a while and see how that goes. So wait, Sam, you asked a question. And what was your question that you were in the middle of, kind of thing?
So really, this is a question about kind of the specifically like
the promoters.
genes
Yeah.
with multiple cancers
Yep.
and serum
Sure,
response
absolutely. Yes.
and how that works.
Yes.
And then the words co-activator and co-repressor.
Oh, yeah,
And how
okay,
those
sure,
fit
why
into
not?
this,
Yep, good.
picture as well.
Let's see, we get this thing going. All right, so we started our course and we're like, okay, so all genes have this kind of organization to them, basically, like that. It's a universal design for genes, and there are always 2 parts. The organization varies quite a bit, and especially it varies between eukaryotes and prokaryotes. So if you showed this to a microbiologist like Rachel Larson or Kat Miller, they would be like, whoa, I don't know. And I'd be like, no, yeah, it's this way. The 2 parts are the output, the transcription unit, and the regulatory region. And they're connected in cis, yeah, like that. Genetically, they're physically connected along the strand of DNA. In prokaryotes, these regulatory regions, they're really compressed. They're really close to the start of the transcription unit. They're actually overlapping the transcription unit. That's fine. There are still 2 parts. They use different terms. That's sad and frustrating for student learning because it's so satisfying to be in our course and appreciate every gene on our planet has this organization, output and regulation, okay? And we learn in eukaryotes, really typically, with very rare exceptions, there's a region that's a regulatory region right next to the start site, and we're calling that the promoter. That's a good term for it. It's also called the proximal promoter. That's a good term for it because it's right next to the transcription unit. It's also called the basal promoter, and that means basic or minimal promoter. It's called the minimal promoter because it's just telling the RNA polymerase where to land. There isn't very much regulation when it comes to the transcription vectors that regulate the promoter, but there is. And we learned some examples. TATA factor is a promoter binding factor that usually just shows up, but sometimes it's the rate limiting step. Like when? Well, like the myogenin gene. That's why it's not getting transcribed, and MyoD helps recruit the counter binding protein, like that, okay? So then there's this other one, and that's the more famous one, because that's usually the one that has the big regulatory protein like transcriptor, like MyoD, and all the other ones we learned, MYC, and all these ones. They bind to what people like to call enhancers. We've learned that enhancer is not a satisfying term, especially when I look at something like the estrogen receptor or other nuclear receptors, because those receptors bind to an enhancer, but it's either going to activate or repress. So instead of calling it an enhancer, a lot of people that are more inside these kind of topics call it a response element. So we use that term some too, like that. Yep. And there's a reason that people still call it an enhancer, and it's because when enhancers were discovered, they were really curious, dramatic things. And why were they curious? Because they could work at a great distance. And we know why. It's just because the distance between the enhancer and the promoter and transcription unit is a tether, a leash. And we're just tethering this powerful protein into this region, like that. Then also you can turn an enhancer around. It works perfectly fine. And so that means like the enhancer is not pointing toward the gene. It's just saying, just do something right around here. So that's that. Then the third thing that's pretty famous is the enhancer can be even downstream of the gene. We're not surprised to hear that because it's just tethered there. And it can even be in one of the introns. It can even be within the transcription unit. So, you know, people are like, oh, those enhancers, whoa, man, they're really important and they're really wacky. But they're not. And all they do is serve as a binding point for this. Then, for most of our course, we just focused on famous enhancers for specific genes, like E2F binds to an enhancer a bunch of target genes. Just a little bit in our course, we introduced the idea that for a given gene, it typically has multiple enhancers. Some can be activators, some can be repressors. It's all the same story, it's just that there's more information to it. And really just one time in our course, it came up where we said, let's just talk a little bit about the complexity of one gene, right? And it's MYC. And we learned, or no, yeah? It's CDK, cyclin. And what we drew is a little bit of a diagram that's a little bit of a maybe false, because cyclins are a gene family. And what we did is we just said, let's kind of draw a picture of a generic cyclin. Because what we learned is that MYC can activate a cyclin. So that could be MYC. But then we learned, and it's kind of confusing until I think you see this. We learned that these other oncogenes, Jun and FOS, they can also activate cyclin. And do they do it through MYC? Nope. They're just a different transcription factors that bind to a different enhancer. And so in some cells, you can have MYC doing the job, in other cells, Jun and FOS, like that. And then we also learned that beta-catenin can do that. So over here, we learned for the first exam that there's an activator, TCF, that's bound to enhancers of beta-catenin target genes, and then beta-catenin, no, so there's a repressor, so TCF is a repressor, and then when beta-catenin gets stabilized by Wnt signaling, beta-catenin is an activator. And so that's just one example of a more typical gene, where it's got multiple stories upstream of it, depending on the cell, the time, you know, like that. And they just vary. And right, it's confusing and complex, but we just have that one example, and we learned about each of those, and you just see how they all come together. So you can think of the cis regulatory region as a way to, and I don't want to kind of use this term again because we used it in a different context, but it kind of integrates or kind of pulls together a variety of different pathways like that. Sometimes some genes need multiple of these to be activated. Sometimes it's kind of an interesting thing, like what is going on if beta-catenin is not here? If TCF is here and repressing, can MYC overcome that repression? And the answer is you just have to do the test. Yeah, so it becomes, you know, it gets to be all, you know, like, you're like, I like sports, and you're like, let's talk about the Celtics, man, the Celtics rock. And you're like, oh, let's just talk about so and so and so and so, right? It's like that, right? It's inside transcription factor biology, right? Transcription factors are awesome, and there's some weirdos that have transcription factor T-shirts, and they're standing there just waiting for you to ask them a question, and I'm one of them. So I would love to talk about this, but that's it, right? I've put the brakes on all through our course, just saying, MyOD and activates myogenin, but there's more to it, like that. That's fine. It's not an oversimplification. We find examples where it's very specific and very straightforward. What's a good example, right? We could take any precursor cell, and if we just force the expression of MyoD, it will activate myogenin, right? So even though myogenin's regulatory region is complex, we can find examples where the story is simple. That's literally true, right? We learned that. What's another one? Right? It's that story of the induced pluripotent stem cells, and you say, I can make any cell an induced pluripotent stem cell, differentiated cells, cell from your brain that's never going to divide again, it stopped, right? I can make it an induced pluripotent stem cell by forcing the expression of MYC, right? So complicated story, but we can find examples to introduce to this course that can lead to framework understanding, right? Understanding of it. But in the real world, usually, there's more stories like that, because think about how complicated our bodies are. There have to be really kind of like these you need more than one to get it done. But it boils down to the same framework facts. And we can find good examples of that. PDGF is a great example under any scab. It's just going boom, 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 MYC, cyclin, and through start, like that. Those examples are kind of fascinating to think about in the context of this course, where I say we're going to have it be kind of focused on cancer. Because cancer, it's complicated, but it also features kind of, accidentally exploiting some of those simpler stories. Like the woman with skin cancer where she's got the translocation, and if she makes PDGF in her skin cells, right? Even though our bodies are really complicated, there are examples that are not oversimplifications where you're like, okay, I get that one. Yep, so that's that. Yep.
And then can we zoom in specifically on the serum response factor story?
Yep. So that's something that's satisfying to see, and it's intentionally set up that way. So we learned for our first exam that the pathway to PGF leads through the PGF receptor, RAS, RAF, MEK, ERK, ELK. And for the first exam, we just learned that, there's ELK, and ELK's phosphorylated. So we got a phosphorylated ELK, and that's going to activate expression of target genes like MYC. And then for our second exam, we see actually all along in our book, nobody asked about it, but there's another transcription factor there bound to the same enhancer as ELK. And it's a team member, and it's called SRF, serum response factor. And together, that enhancer is actually referred to typically as SRE, the serum response element. And serum is just the liquid component of blood, and in a baby, there are lots of growth factors in the blood. The baby's growing a lot. And so scientists use that serum just a way to stimulate cells in a dish, because it just has good general growth factors. If you remove the serum from the dish, the cells will stop growing. Even though they have good oxygen, they have good nutrients, they need the growth factor to go through starch. So it's a satisfying addition to our story. Yep.
And so the serum response element is the overall dimer. It's not the genetic, because usually we say response element to mean the section of DNA that's bound.
Yeah. Do these bind to the dimer? I don't know. Probably not.
Okay.
Yeah.
And then this is a different story where both of these are necessary.
Sometimes not.
Wait.
Right. So we learned about it, and we could drive into it further.
Sure.
Because almost for sure, sometimes ELK doesn't need SRF. Sometimes SRF probably doesn't need ELK. Like that, where, and that's because our bodies are so complicated, and we haven't oversimplified it. There would be examples where, like under the scab, I think you probably don't need SRF. I don't know. Maybe it's there. He was hanging out all the time, you know? But it just dives into it beyond the basic principles to you know, significant direction, significant path, but one that starts to confuse and cloud the fundamental principles. You know, so I want to make that really clear. It's not an oversimplification, it's an establishment of the framework principles. And we pick examples that are true and not an oversimplification at all, but are really generally simpler examples than the more common examples in our bodies and nature, which is, oh, you gotta have another one. And don't feel like you've been shortchanged at all or anything like that. This is how you have to get into something like that, right? Where the car mechanic doesn't tell you about every part of the car, they say this is an important part, it's called a tire. You know, and you're like, okay, got that. And they're like, I didn't mention the lug nuts, but we're just focus on the tire, you know, like that. So it's good to ask questions to find out what more do we need to know. Yep. Yes.
And then the words co-repressor and co-enhancer.
Yeah.
How would either of those reply to SRF?
I don't think so. I think I'm sure SRF and ALK both have DNA binding domains and bind directly. So let's just use an example of that, just to clarify that. And so we'll go back to the examples where we learn these terms. So one example is the estrogen receptor for our first exam, and remember that we're already thinking about the final exam. So this would be the estrogen response element, and it's going to have the estrogen receptor bound to it. So that's estrogen receptor, and it's going to have estrogen as a regulatory ligand, right? And we learned that the estrogen receptor has a zinc finger DNA binding domain. It binds directly to the DNA. It's a transcription factor. It's a protein, and all transcription factors are proteins, and this one has a DNA binding domain. But then we learned that there are these other molecules that bind to it that can regulate its activity. So we can have the histone acetylase or the histone deacetylase binding like that. And those don't bind directly to the DNA. So some people would say we could call these co-regulators, and that's fine. In our course, we can. It's fine. They fit the definition of co-regulator, which is co-regulators are transcription regulatory factors, so they're transcription factors. And what's the deal with a co-regulator? Co-regulator is a transcription factor with no DNA binding domain. So it's going to bind to another transcription factor to be tethered, right? So you got the tether, the leash, you got the dog, and then you have the little monkey riding the dog's back. And the dog is super nice. The monkey is super bitey. And the monkey is riding on a saddle on the dog's back. That's a co-regulator. And a co-regulator is either going to activate or repress transcription. So people say co-regulator, but more often they just go right to co-activator or co-repressor. And who can think of a co-activator that we learned about in our course? And if you answer this, you don't have to take the exam on Thursday. You get 100. Yes, Joseph.
Is RB one?
No. You have to take the exam, and the most you can get on the exam is 50. Yeah, no, you play. Yeah, no, you play to perish. That's the way this game works. It's just like TV, right? Where you're like, you play.
Is beta-catenin a coactivator? Because it binds
Okay. Yeah.
to the,
All right.
I mean, I get
See you
100.
next Tuesday. Yeah, sure. Why not?
Wait, really?
Sure. Why not? Yeah, sure. Don't come on Thursday. Don't worry about it. Yeah. Yeah, and eventually you'll probably end up talking to the dean about it. You'll be like, there were witnesses. There were like 20 witnesses.
It's recorded.
It's recorded on the World Wide Web. Yeah, beta-catenin is a co-regulator. It binds to the back of TCF, and it's a co-activator. It's going to activate it. Lydia.
But does that make June and Foss co-regulators because they can work together?
No, both June and Foss both have DNA binding domains. So they are binding together as a dimer, together teaming up like that. Yep.
Can we talk about why RB isn't one? Because I thought it.
What is RB?
It's a transcription factor that binds to the E.
Too many words. What is RB?
It stops RNA.
Too many
polymerase.
words. You're out. Yeah, you didn't even buzz in anyway. Yeah, Salima. Okay, yep, Lydia. Okay, who wants to say something else? Rustam. Is it a co-activator or a co-repressor? It's co-repressor. Yep, that's an example we learned of that's a good co-repressor. RB is a transcription factor with no DNA binding domain. It must be a co-regulator. It's a co-repressor. It binds to the back of the E2F transcription factor and represses it. So even though, I helped guide Rustam, also does not show up on Thursday. Nice, right? We'll have more room for your exam on Thursday, right? Yeah, okay? All right, let's just jump ahead. No, let's not jump ahead. Those are good examples. Yep, yes?
So acetone acetylase, is that an activator and deacetylase is a repressor, or are they?
Yeah, so we can call them that. It just, because it fits our definition. They're great, so like that. I would just say that, out where the people really talking about them, they just don't use those words, because they're just like, it's an enzyme, like that. It's great. But as far as the strict definition of the word, sure. Histone acetylase is a coactivator. Histone deacetylase is a co-repressor. Yep. Yeah. Other questions people have? Let's see. I think we're doing great.
So, can you kind of just talk about MCM and what we need to know about that for Thursday?
Yep. And so I don't have that figure, but let's just talk about MCM. So. Let's just talk about why it's called MCM, because sometimes that's kind of a satisfying thing, and it's called MCM for a reason, and it looks like this. Here's a yeast. It was studied first in yeast, and here's a yeast, and it has chromosomes like that, right? So we learned about that. It has some chromosomes, and we learned that when the yeast goes, and any of these cells go into S phase, they're going to copy the DNA, and they're going to use DNA polymerase to copy the DNA. And there's a lot of DNA. So when we see yeast or any cell go into S phase, the DNA polymerase binds to many sites along the length of it, because we got to get this thing copied. And everywhere it binds is called an origin of replication. How did scientists discover origins of replication in eukaryotes like us? In yeast, they just took a piece of a chromosome and they put it in the yeast cell, and they would ask if that piece of DNA would get maintained during the cell cycle or would it get lost. And what do you think the DNA needs to have to be maintained? Let's just keep playing for this and see if we can have nobody here. No, you are not coming on Thursday, so no. Yeah, you're out. Yeah? Yeah? What does this need to have to be maintained in the cell? You don't have to frame it as a question like Jeopardy, you can just go for it. For this piece of DNA to be maintained, it needs an origin of replication, or it wouldn't get replicated. Okay? And so it's got to have an origin of replication, because then DNA polymerase will be guided there to bind it. See that sense? Right? It's an experiment, and you're just like, we need to figure out what it takes to get the DNA copied. Well, let's just put another piece of DNA in and see if it gets copied during S phase. And it's either going to get copied or not. And if it gets copied, it's got an origin of replication. Like that? So the scientists in yeast, who are all super cool, call this a mini chromosome. And MCM stands for mini chromosome maintenance. Because you need an origin of replication, and then you say, all right, now we have a mini chromosome. Now we can identify everything that needs to be used at the origin of replication. And if we have a mutation in MCM, it won't be maintained. Because MCM helps the DNA polymerase find those, right? So the DNA polymerase needs to find them, and one of the helpers is MCM. So before the DNA polymerase shows up, MCM shows up. Mini chromosome maintenance. And it's there in all our cells all the time. During S phase, we need a lot of it. Just let's go, right? So let's just draw a picture of the MCM gene. For us, if we had to draw a picture of the MCM gene, we'd look like this. We'd be like, so there's the MCM gene, and I bet that it has E2F, transcription factor, binding an enhancer, and I don't know this is a fact, but it's for sure. E2F is trying to get the cells going into S phase. And what do we need during S phase? Lots more of the proteins needed for S phase. What examples did we use? DNA polymerase is a protein, and we need more of it. So E2F is bound to the enhancer of DNA polymerase. Let's crank it up. We need enzymes that catalyze the synthesis of the nucleotide precursors A, G, C, T. We need a lot of those to copy the DNA. E2F is bound to those. We need the components to recognize the origin of replication. E2F is bound to the MCM gene. So that's the first thing that we want to emphasize about MCM is we're going to need a ton of that protein. So we're going to crank it up with E2F. Then we just add to that, in our story, we know that this is one of the things that retinoblastoma is normally bound to. And it's saying, nope, nope, nope. We're not going there. The light is red. And this is what it means when the light is red. It's not a green light, red light in your cell. It's just biochemical and genetic situations. And this is one. Probably not going to go into S phase if you don't have very much MCM. So we'll just keep that, down low. Okay? Then the next thing we learned, and in the figure in the book that shows a lot of detail of the origin of replication, we just picked one. And we said, so we get the protein made, great, and we get lots of it made. Then we have a second level of activation, which is MCMs are a target by the CDKs at start. So, we're going to get them made, and then we also are going to add a phosphate. And that's going to help activate the MCM activity. So it's kind of a double whammy, and it goes back to that thing of let's really turn the light green. Make a bunch of MCM, and then let's phosphorylate it because it's more active when it's phosphorylated. So two pieces to that. And, we kind of talked about it briefly in class, but this clarifying question makes it more solid for, it won't be the hardest question on the exam when I say what are, to explain two features of MCM that, came up for why we wanted to learn about it in regulation of the S phase, status. Yep. Yep.
Can we switch gears? I have a couple questions about the small inhibitory proteins like P27. I was wondering what activates it because it seems
yes,
like
but wait, let's just do this. So there's a little piece of more. We just talked about retinoblastoma, and we see retinoblastoma functioning as a transcription factor co-repressor of E2F. And E2F is just let's go into S phase. Let's copy a bunch of DNA. Let's just fit this little piece of new material that we still want to cover today. Let's fit this in here because it includes retinoblastoma in a real satisfying way, that will kind of underscore what we just talked about. So let's look at that, okay? So it goes back to this, where in our book, it wants to talk about viruses and the story of viruses in cancer. And we've been learning about retroviral oncogenes. And those are a special kind of mutation of retroviruses. Retroviruses in general don't cause cancer, they just cause different kinds of diseases in all kinds of different animals, like that, right? And a retrovirus is a specific type of virus, a certain category of virus. So we want to talk about another example of a virus that contributes to cancer, not in that direct way of having a host gene that causes cancer, but in a more just natural way in the world, okay? Because our book talks about them too, and one of the reasons it does is because these are some of the most important ways where scientists got a chance to start learning about the molecular basis of cancer. Oncogenic retroviruses in the 1970s. Also chromosomal translocations, because they saw those. They saw that Philadelphia chromosome and the MYC and the woman with cancer. They had seen those from at least the 1950s. Yes?
Oh, just stretching my hand.
Oh, me too then. And gene amplification. Another thing that was an early feature where they're like, that is a weird detail in that cancer, where it's got lots of MYC, right? And so here's one too. And there's a huge bunch of stuff about it. There are several viruses that can increase the likelihood of a person getting cancer. And we're going to talk about one of them. And RB is part of the story. So, in looking at these viruses, we just have to talk about viruses for a couple of minutes. There's lots of different viruses, and they get categorized by the type of DNA or RNA that's in the virus before it infects the cell. So you don't need to look at this in detail, don't need to be able to see it in detail, but there are really sort of 7 categories of viruses that infect eukaryotic cells on our planet. And they really got categorized based on what the virus has inside. So over at this end are retroviruses. And that other arrow was just a little typo. So here's our retroviruses over here, group 6, and they have an RNA genome. And the first thing they need to do is they need to copy it. They need to reverse transcribe it. Down here, it says, over here, group 6, it says the virus has to contribute some activity. And for these viruses, the virus has to bring a gene that will code the reverse transcriptase enzyme to copy it from RNA into DNA. Over here, these are DNA viruses. Do they have a reverse transcriptase? No, they don't need that. So they're very different viruses, just different categories of viruses like that. So we're going to move over here and talk about a DNA virus. Next thing is, when a virus comes into a cell and infects a cell, what's going on? There's one thing that's going on, and that is the virus is just going to use help from the cell's biochemistry to copy its genome. It copy its, if it's RNA or DNA, it's going to use help from that and make a bunch of viruses. It's using the cell to help it. So the virus has to contribute some genes, like the reverse transcriptase, but it's using a lot of host genes. So for reverse transcriptase, we learned that it goes into the cell, copying the DNA, inserted, and then it uses the cell's RNA polymerase to copy RNA, transcribe RNA, and that's how it gets its replication. What's this one going to do? It's going to come to the cell and it's going to say, I want to make this cell copy my DNA a lot. And how's it going to do that? It's going to manipulate the cell. What would be one good idea? Inactivate retinoblastoma. Right? All the machinery cranks up. It doesn't care if the cell goes into S phase. It just wants machinery to copy its genome. So that's just a simple example. Viruses are very, very simple. They're just going to infect, and if it's a DNA virus, they're going to be like, I don't really care if you go into S phase. I do want to get access to the stuff that's in S phase. I would like to see a lot more DNA polymerase here. And so they just get mutations that lead to that, their success, right? So we're going to talk about a virus that's over here, and it's called HPV, and there's a good chance you've heard of it because it's a famous sexually transmitted disease. It causes genital warts. So here's the situation. It's super common. Most people that get it as a sexually transmitted disease get better really quickly. And they're all healthy and they're done with HPV. Some people have the infection linger. It doesn't get completely better. As a result, those people can get over time genital warts, which are just little bumps. Why are they getting those? And the answer is those cells are growing too much. Are they cancer? No. But they're growing too much. Why? Because the HPV is pushing for the cell to help it copy itself. And in doing that, it tends to push the cell through the cell cycle. The virus doesn't need that, doesn't care about the cell and the cell cycle. It's just trying to push, okay? So when we think about what is the HPV bringing in, it's bringing in stuff either that it needs, like over at the retroviruses, reverse transcriptase, or bringing in stuff that will help it take advantage of the cell, right? And so this is just a list from our book. It used to look like this, a little bit prettier in previous editions. And then there's a figure that I guess just isn't in the edition anymore. But it's so freaking satisfying to see this, because one of the genes that HPV brings in during any infection is a gene that inhibits RB. Why would it be doing that? Because then E2F is active. Does the virus care if the cell's in S phase? No, it doesn't care anything about the cell. The virus cares about the fact that in S phase, the cell is making the machinery that the virus wants. DNA polymerase, AGCT, like that, all kinds of stuff like that, right? So it just pushes it, right? And it's blocking the tumor suppressor, right? So people who have a lingering infection have this vulnerability stuck, in their body still. And it could be for years where they're just vulnerable to the mutation, right? Because already the retinoblastoma is being inhibited. See that? So then just one more is also super famous, is another gene that comes in with the HPV genome is going to also degrade P53. And remember, P53 can put the brakes on the cell cycle. Does the virus care about the cell cycle? No. But if it blocks P53, remember P53 is a transcription factor that activates P21. When the cell's infected, it's like, this is trouble. I'm copying a lot of DNA, I'm making a lot of mistakes. We should block, we should stop. And if it's really problematic, we should go through apoptosis, right? So what an epic target for a virus that needs to copy DNA to block. And this is what normally happens to P53 in the absence of DNA damage, is it just gets destroyed. So here's a virus that comes in, it makes its little product, and it starts destroying P53. There's no chance that P53 can help anymore. So if there's DNA damage, need repair, and there will be, because viruses do not care about accuracy. They're just like, let's make a lot. We're going to make thousands of viruses, and if some of them are wrong, who cares? Let's roll. Let's party. Yeah. This is like a friend that comes over to your house, right? Your parents' house, right? And they're like, I found the key to your parents' liquor cabinet, so let's go, right? It's just wicked trouble, right? Again, most people get over HPV infections, completely eliminated from their body, they're fine. Some people have it linger, and they have this vulnerability. That's where the genital warts come from. It's just a slow growth. It's not cancerous, but it's like, it shouldn't be growing like that, right? Okay? So just really satisfying how it fits into our course, right? We learned about P53. We understand very well why the virus would evolve to degrade P53. And we go back to that, and we learned about retinoblastoma. We're kind of experts on that. It's just so satisfying. It's a tumor suppressor, right? So E2F target genes, right? We're just going to, you get rid of retinoblastoma, you just give these the green light. And we've just been talking about the hundreds of things that those activate, and it's all about S phase. And again, it's a DNA virus. It wants to have the DNA replication machinery rolling like that. So rolling the cell cycle. So E2F family members, we learned about them, right? Cyclin, CDK, check regulators, DNA repair, replication protein like MCM. Yep, okay. So then a little bit more about HPV, Wikipedia page, because it's a human sexually transmitted disease. So for our own medical health, we should have some awareness of that. And that's what it talks about. Genital warts, Wikipedia page, like that, you can learn about that. And right, this is not a course on your, medicine, but, there's an example. Then there are specific kinds of cancers that are associated with HPV. What kind of cancers? Well, cervical cancer, not brain cancer. The infection takes place in our reproductive organs. So they're going to be really focused on that, especially, stuff associated with sexual activity, And then there's vaccine. And it's famously controversial because the vaccine. But if people get the vaccine before they're reproductively active, they're not going to get cervical cancer in their lifetime, because almost all cervical cancers, and you've heard about those, almost all of them are caused by lingering HPV infections, like that. And that's that, right? So that's a part of our chapter 19. And the book just kind of deleted that part out, because it's always trying to streamline and make it, like that. But man, what a satisfying thing to have, in this chapter, because it shows you how we have a framework of knowledge, and we can add to that, in different contexts, stuff like that. So I want to just tuck that in. Great. And then we'll go to this question. Yeah, or anybody questions about that? Yeah, so a little bit new knowledge for an exam that should be chapter 19 information. The book talks about viruses, and it's just really valuable to appreciate that there are other examples of that, and so forth. Yep, yes.
Is it CDKs that phosphorylate RB, or is it CDK and cyclin together that phosphorylates RB?
It's, so CDKs are cyclin-dependent kinases. So it's always going to be, when CDKs are active, they're always going to have a cyclin bound to them. So RB normally gets inactivated by getting phosphorylated by CDKs. Yep. So other questions about HPV type topics, or the the cancer and viruses topics? Yeah. That I like that, because again, it's kind of a thing where you see how we have this framework of knowledge and you can get going on it. And then molecular biology is always like this, where you're just like, oh my God, oh man, I got to take a walk, right? I just had to take a walk down by the river, and man, because it's just, amazing when you see at that biochemical and genetic level, the reality, the nuts and bolts, right? Like that, right? Here's a great example of that in real life. Great example. So my kids are older than you, and when they were little, my wife drove them around in a minivan, and I drove a car to USM. And I didn't drive around in that minivan hardly ever. And she doesn't know anything about cars, right? And so she would take it to get serviced. She was kind of doing the service on that vehicle. And one day she said, yeah, I was driving it, we'd gone in and rotated the tire. Driving it, and the front end's really wobbly. And I was like, okay, I'll check it out. So I got out and I just drove down the driveway. The front end was wobbly, and I took off the hubcap, and the car has 5 lug nuts, and 3 of them are sheared off. And the tire is just on the front of my family's wife and 2 kids is riding on that. And the people who rotated the tires at the mechanic place, they just torqued them on too tight, and it just ripped off the lug nuts. Yeah, And so it's this vulnerability where you're like, oh my gosh. I called them up and I said, come tow this to your place, fix it, and get it back here, and you're not going to charge us anything. Cancer's like that. When you look in the details of it, you're just like, oh, and you see the molecular details of it. That's just so difficult, because you're like, oh, that RB is so important, and the virus is blocking it. Yeah. Okay, so that's that. Other questions people have?
Okay, so a couple questions about the small inhibitory proteins.
Mm-hmm.
So at start is P27 usually bound to the cyclin and the CDK, and then we have to remove it in order for the CDK to be active?
That varies.
Okay,
So
that varies.
sometimes the start is dependent on a small inhibitory protein that needs to get removed. Yep. Other times not. In some cases it's only because there's a problem like DNA damage, and so those small inhibitory proteins get activated to block the cell at start because of a problem. Yeah. Yep.
So I was trying to figure it out, and I was trying to figure out, so I was like, okay, well, if you have to get the light flickering, but it's being blocked by the P27, what allows for some flickering? And I guess my brain can't understand, if you're blocked, how are you going to get some flickering? And I think it goes back to, you have to get, the E2F protein.
Sure, yeah. Lots of these questions, and they're great, lots of these are kind of probing that further knowledge.
Okay.
right? Sort of a detail or more complexity, like what are some of the features of this? And we could go there,
Okay.
but it just is gonna, it would start, kind of confusing, and like, I'm
Okay.
starting to unremember some of the foundational things. But yeah, remember that when we started the chapter on cell cycle, we said, we're gonna go over to the G2 to M transition, because there's a very nice switch there. And then we're gonna go over to G1 to S, which is the one we care about. That's the one their cells are gonna pause in, and it's complicated. And so we've done a really good job framing out the foundational principles of it, right? But it's, we're, at some point we'll stop and we'll say, we're not gonna get more complicated in trying to explain the nuances of when is, P27 really holding it back, and why are the other things green? Why is the light flickering?
I just didn't know it
Yeah.
was like stage 1, it was
Yep.
always there, and then we removed it, but
No,
that's okay.
no, definitely not. So for these small inhibitory proteins, yeah, lots of different reasons. We picked P27 just to get the principle of how we could have this positive feedback, where the light is going to turn green. So that when the CDKs get activated, the light starts to flicker green, and some of their activity helps turn it fully green. For example, they'll phosphorylate at P27 and get that removed. So that's just the whole story with P27, and scientists know a lot more, and there would be examples that would resonate with the stuff we learned, but that's great. And then P21 is that's just our thing for DNA damage checkpoint, that inside of the start control point, there is this additional checkpoint. And that's a nice thing to learn about, because it's like the light is green, we've done everything we need to do, but we're waiting because there's DNA damage. Yep.
So is P53, I know P53 activates P21, which is a small inhibitory protein. Is P53 a small inhibitory protein?
No.
No, okay.
No, yep, yeah, and I mentioned when we started talking about them, lots of times biochemists will name a new protein based on just its size. So they're just saying the size, because that's all they knew about it. So P stands for protein. We always, that's their P, and then the number stands for its molecular weight size. And so a small protein is always a relative thing, right? Do you have a small dog? Well, that's relative. Is Maisie a small dog? In
Right,
some
Maisie
cases,
says yes.
right? But yeah, but in general, it's kind of a loose term, but a 53,000 Dalton protein is not a small protein. It's a relatively bigger protein. So, but definitely P53 is not a small inhibitory protein, and we could go with the size, but then more importantly, we go with its function. It's a transcription factor. It's got nothing to do with binding CDK cycling, which is what small inhibitory proteins do. Yep. Right?
Another question about P53. We mentioned at some point that there was a linkage between the P53 and the BCL2 family. What is that? I couldn't find it
Yep.
in my notes, so I
Yeah.
was lost.
Well, we learned that P53 is a transcription factor,
Yes.
and it is going to activate P21 gene, and P21 is going to be a small inhibitory protein.
Yep.
So why would we do that? And the answer is that if there's DNA damage at start, that damage will lead to the stabilization of P53 protein, and it will get phosphorylated, and it will bind to the enhancer of P21 gene.
Yep.
And then we learned that there's a timer mechanism, that CHEK2 kinase just keeps phosphorylating it slowly, and when P53 protein gets hyperphosphorylated, it starts binding to other target genes,
Apoptosis.
pro-apoptotic genes.
Okay.
So we don't need to learn the name of those, but it's going to lead to the death of those genes
Okay.
by apoptosis, the death of those cells by apoptosis. So we're going to give you a chance to repair your DNA. You don't fix it, you're out of here. Yep. And so it's nothing to do with BCL2. What it does is it activates genes that are pro-apoptotic genes, code for pro-apoptotic proteins, that are members of the BCL2 gene family.
Perfect.
And that's where we say we're going easy on that. BCL2 is an anti-apoptotic protein, and we learned about it in non-Hodgkin lymphoma and the chromosomal translocation. So we kind of dove into BCL2, And then we learned that there's also a gene family. And the gene family is called the BCL2 gene family because BCL2 is so famous. Within the gene family, some of those genes code for proteins that are pro-apoptotic, some anti-apoptotic. Yep. Yep. So let us, yeah, Lydia.
Just like to clarify that, so it doesn't necessarily like stop the caspase in that, because it's pro-apoptotic and anti-apoptotic, so I have in my notes that it represses that it blocks the cell death, but that's not necessarily true, it's promoting it in this case.
Yeah, so when we say something is anti-apoptotic, it's inhibiting the activation of the caspase enzymes. The caspase enzymes will kill the cell. They'll flip they'll activate the scramblase and flip the phosphotylserine and so on and so on and so forth. And so all the BCL2 gene family member proteins are all acting upstream of caspases, and some are on pathways to help activate it. Pro-apoptotic will mean activating caspases. Some are going to be upstream and inhibiting it, and those are anti-apoptotic. And so BCL2 is actually a couple of steps upstream from caspase. It never touches caspase directly, but it's regulating the caspase and keeping it off. Yeah, Cam.
So P53 just the transcription factor for the KIP family?
Transcription factor that what?
The KIP family, which is P21,
No.
P27.
Nope. No. So the KIP family are small inhibitory proteins. They function by binding to the CDK protein, and they inhibit it. P53 doesn't bind to the CDKs. P53 is a transcription factor that binds to the DNA upstream of target genes. And one target gene of P53 that it can activate is P21. P21 is a coding gene. It codes for a protein, P21 protein, and that binds to the CDKs. Yep. Right? So P53 is not a small inhibitory protein, and it doesn't function like them. P53 activates expression of a small inhibitory protein. Yep.
So in the pro-apoptotic pathway, does CHEK2 just make more P53, or is that hyperphosphorylated?
It's phosphorylating it. It's phosphorylating, so it's activating it. It's helping stabilize and activate it, and then it's also going to eventually hyperphosphorylate it and get it to help kill. So first, it's going to cause it to bind to the P21 gene and activate that in small inhibitory protein expression, and then eventually it's going to get hyperphosphorylated and bind to these pro-apoptotic members of the BCL2 gene family.
Yeah, and ATM is also part of this pathway, and it says it activates CHEK2. Do we care about that?
No, that's just the upstream, more details about how is the DNA damage interpreted. So ATM is binding to the DNA damage complex that is finding the DNA damage and saying ding, 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 and it's communicating, it's activating CHEK2 through ATM. Yep.
And then CHEK1 inactivates CDC25, which prevents the removal of inhibitory phosphates.
Yeah, and it's easy to distinguish
CDK1.
between CHEK1 and CHEK2 because we learned that CHEK1 plays a role at the G2 to M transition, and CHEK2 plays a role at the S at the start, or G1 to S transition. Yep. Yeah. So let me show you a little more because I said there are these 8 things, and we won't have a concept chat today, but we'll just roll through this list. And the book has over the last couple editions, it used to have this list, and it used to frame a lot of the chapter 19 based on this list. And it's just satisfying, it's a satisfying way to organize a little bit of our knowledge of cancer. And so this could be on the exam where I say, briefly describe 4 of the 8 things on this list, right? And some of them we're already familiar with, but let's just roll through this because I just think it's satisfying. So you don't need to scramble to write them down terribly because remember we're trying to record this, so it'll be available on YouTube, but also I'll just show it again and again. So here's the thing. The list goes like this. The first ones are true about cancer, and they would be true in our bodies. but they were really seen clearly when scientists took cancer cells and they grew them in a dish. So in a sterile dish in a lab, in liquid nutrients, they would raise these cancer cells, and they found out these features of them, right? Okay, so the one, uncontrolled proliferation, we got that, right? They're going through S. They're just gonna keep going through S. Here's how that's seen in a dish. And let me just show you, I think I have a picture of maybe, cells in a dish. And so uncontrolled proliferation, something that's true of a normal cell in a dish is it's gotta stick to the plastic at the bottom of the dish. It's in liquid, but it's just sticking to the plastic. And it grows and grows and grows, and when it fills up that layer, so each cell is stuck to the dish and it can't fit anymore, normal cells stop. They're like, we've done this, whatever it is we're doing, it's kind of weird environment, but we're done with it. And all cancer cells don't stop. Uncontrolled proliferation, so we know that as uncontrolled S phase, and it includes this thing of they're just not being normal. They, and you know, in our bodies, that's what happens, right? The cancer is just gonna keep rolling. You know, whatever neighborhood it's in, it's just gonna keep rolling forward. So that's the first idea, again, in a dish, okay? Then, reduce dependence on growth factors. So that's something we're also comfortable with because we have that example of the woman's skin cancer. Her skin cancer is caused by PDGF problems, but she doesn't need the normal, the normal pathway for the growth factor anymore. We saw ERB mutation, the urotransferrin receptor is mutated, it doesn't need the growth factor anymore. We saw those RAS mutations, they don't need growth factors because the signal transduction pathway is roaring. All those examples are that example of reduced dependence on growth factors, you know? Then this idea of capacity for unlimited replication. So again, we're talking about a dish, and what we said is that the cells will not stop, and they'll just keep growing, and they'll keep growing forever. So in a dish, in this setting, one of the hallmark features, one of the number one features of cancer cells is they'll live forever. They're immortal. If you took our cells, normal cells, and put them in a dish, they would stop growing. If you diluted them, they would keep growing. They'd divide more. If you dilute them, you don't give them more space to grow, they'll divide more. But they won't grow forever. And interestingly, if you take normal cells from a baby, it will go through a lot of growth before it dies, because each of the cells is mortal. And if you took cells, normal cells from an old person, they'd only divide a few times before they would be done and die. In contrast, you could take cancer from a baby or an old person, and it's never going to stop dividing, ever. So a curious feature of cancer,
Is that the Hayflick value?
it is. It's called the Hayflick limit. And we don't need to learn that term. Just the concept of the cells are immortal. Never stop dividing. Immortal. And in a dish. And then, so uncontrolled proliferation, reduced dependence, capacity for unlimited replication, and then this reduced cell adhesion, reduced dependence on cell adhesion molecules. What that looks like in a dish is back to this drawing. So the normal cells, they have to hold on to something. Usually when people, no, I won't say that. They just have to hold on to something. And if they can't hold on to something, they'll stop growing. Cancer cells, they can hold on to something, but if there's nothing to hold on to, they'll just keep dividing. The reason is because on the surface of every cell, there are some molecules that stick to other cells normally. And cancer cells just have lower levels of those. We see that in cancer when it spreads. The cancer can metastasize, and what's going on there is the cancer has just lost that dependence to be touching and holding on to other cells or plastic. It just is like, I don't need that. And that's an example of how the cancer progresses, right? It's local, it's growing locally, and then when we're worried, it might spread. When it spreads, it's because it's having a decreased dependence on these cell adhesion molecules. So that's the first 4 things on this list. And I just sort of summarized them there because that limit thing is called contact inhibition. So when they make contact with other cells, their cell adhesion molecules are like, okay, I think we should stop. And they just have less dependence on that, touching other things, holding on to other things. And, but that's that's our first half of our list. We're doing great. And it all comes from just taking the cancer cell out and looking how it behaves in a dish. And basically all cancer cells behave that way in a dish. So really curious, right? Because like that immortality, bad, right? So there are features like that. And they fit in there. Then these next, some of them are new. The last one's fairly familiar, right? So we got that one, failure to undergo apoptosis. We learned about that and how BCL2 plays a really important role in inhibiting it. And some cancer cells just have too much BCL2 activity and they can't go through apoptosis even though they're messing up. These 3 are just kind of curiosities that are back in the body. And so I have some slides for them, but let me just kind of briefly talk through each one. So secretion of proteases. So we learned proteases cut other proteins. Cancer cells grow and they invade. They spread into other tissues. One thing that normally limits cells spreading is that extracellular matrix mesh. All our cells are surrounded by this mesh. And the cancer cells are just chewing that up. And that just lets them spread more like that. So that's something that's really common in tumors. In solid tumors, they're just getting bigger and they're pushing into other tissue. And the other tissue is kind of like, hey, buddy, stay in your lane. And the cancer is like, I'm just going to be cutting up proteins that are in my way. So that's just that. And then the cancer tumor is getting bigger. Angiogenesis is new blood supply. So it needs to get some more blood supply, some new arteries and veins growing into that. So cancer cells also stimulate this angiogenesis, blood vessel growth. One of the reasons that these are listed here is because they're targets for cancer treatment. A long time ago, a scientist realized that maybe we could inhibit new blood vessel development and we could literally starve the tumor because it wants to get bigger, but it needs a blood supply. And in a grown-up, there's not a lot of new blood vessel growth happening. So we can just inhibit it. And now there are some cancer drugs, some chemotherapeutic pharmaceuticals that just function by inhibiting new blood vessel growth. And we're trying to slow the cancer down just by starving it from the nutrients that the blood vessels bring, starving it from the gas exchange. Yeah, like that. And then the next one is one that we learned about, and they say abnormal differentiation. And that is an example that the book has a figure of, so I'll show you a figure there. So these are just back in the body and some features that are kind of famous to hear about for cancer biology and cancer research. One of the reasons to list them is because they're still about cells and proteins, but they're also a lot more about like the tissue and the setting, the tissue setting for the cancer in our body. And I like to include that because we spend so much time really focused on what's happening inside the cancer cell, what's happening around the cancer cell. So here's just some figures from that, and I don't even have figures from that, but I have a figure from this one. And what this one shows is something that we'll be able to appreciate. And this just shows that this is the stem cell in our bone marrow. And I think this is still in our new book, but it just doesn't really pin it down. Here's the stem cell in our bone marrow, and it's dividing asymmetrically, and it's making the precursors to our blood. So we learned about that and how many it's making all the time. So they divide, and we got a precursor cell, and the precursor cell divides. And then remember that we say the cells will stop dividing, but if they get activated, they can divide more. So a lot of the immune system cells that we have in our blood are unusual because they've differentiated, they're playing their function, but they are ready to divide more. And this just says that there are some cancers that actually harm the cell at that stage. And what happens is they get the cell to never fully differentiate. They keep the cell in that state where it's like, I can divide more. And so they don't have mutations in the oncogenes or the programmed cell death genes. They just have mutations that block the cell from fully stopping. And the cell just keeps dividing because it's like, I am a specialized cell that is not like a muscle or a brain cell. I need to be ready to be activated and divide more. And so it just gives another example of just a little bit more of the complexity of the story of cancer in our bodies. Yep. And then there's the, the failure apoptosis one really filling out. So I just wanted to cover these because, again, the book is trying to put the chapter back into the context of cancer biology, and like the cancer is in our bodies. And so, we spend a lot of time saying, oh, this cell is cancerous because it's expressing MYC. But also, the cancer is in that cellular tissue environment. So we should just be aware that the cancer is having problems in some of the other areas of being a good neighbor, good cell for us. So that's that. And again, it's not well organized in our book. But once you see the list, you see the book is talking about these, but it just doesn't really pull them together into a thing like, here's a framework, and also the idea of why are we going across these? And so there are really two reasons. One is scientists study cancer in a dish, and it has these qualities in a dish that are really interesting and universal for cancer. And then the cancer cell is in the body, and it has these characteristics where it's going to need to get moving into other tissue. It's going to need to be in a tumor. They're going to get some new blood supply. There's some situations where it's just not going to fully differentiate, like that. Some situations where it fails to die. So that's that list. And with that, we're good for stuff that I wanted to talk about from chapter 19 for our exam on Thursday. And again, this would just come up with saying, we learned about some features of cancer, list of 8, and describe 2 of them or 4 of them or something like that. Yep, yeah, so it's a list and an explanatory list, brief explanation. So now we can go back to questions because we won't have a concept chat and we have another 5 minutes to party. So I think you had another question.
I did. So a while ago we were talking about cyclin proteins, and then
I'm just doing this because this is what you people do. I don't do this, but when somebody's talking to you, you start doing this. So yeah, go ahead. I'll just be texting while you're asking your question.
Do I do that to you?
No, I'm not accusing you specifically. I'm accusing all young people of being like this.
Okay, so
Yeah?
we said that the geneticists
Yeah?
they found that the
Oh,
CDCs
yeah?
for the cell division cycle.
Sorry, what to see? What? Sorry. Okay, I'm done. Yes, sorry. I wasn't mocking you. I was mocking your generation.
I just have a question because we said that
Yes.
everything that the geneticists made, they called the CDCs, which was the cell division cycle plus a number.
Yeah.
But then we called it CDKs for the rest of the time.
Right.
Is there a difference between CDC and CDK?
Remember how people stumbled when I showed this slide and I say, over there are the mammals and their programmed cell death pathway, and there's the word caspase and the word BCL2. And over here are these simpler organisms where those orthologs of those were all originally discovered. And then there was this column over here for the people that studied down in Boston and found all of them in this real simple work. Found them genetically, and they found them fast because they had a real simple system. And there were SED3, SED9, and people asked, what about that? We don't need to learn those terms at all. Instead of calling it SED9 anymore, they would just say that's a caspase, like that. So the same is true for the cell cycle. The people who originally discovered orthologs for many of these stories that we have for the regulation of start and regulation of G to M transition, those people originally studied them in simpler organisms. Yeast a lot, genetics in yeast a lot, and also I mentioned biochemists in frogs, like that, so simpler systems and situations. The yeast people were geneticists, and geneticists tend to pick a class, like we're screening to look for cell division cycle mutants, so we'll call them CDCs, and they just number the genes, because they have no idea what they are, and they just number them. So I'll just say this, that what we know of as CDK1 today is an ortholog in all eukaryotes, for sure. Plants, animals, single-celled organisms, like yeast. And it was in yeast where it was first discovered, and it was just one of all their mutants, and it was called CDC1. No, 2, CDC2. And you're like, why did I say that? Because it's just confusing. So the CDC terminology is out the window,
Okay.
with the exception that people still call CDC25, CDC25 phosphatase, because it's so cool, and so many people back in the 90s got CDC25 tattoos that they're like, I would like to keep this name. But all the other CDC number names got moved on to CDK1 and cyclins and so forth. Yep.
Can I ask one more question about being boo?
Oh yeah, boo.
So we said that
no,
there
we
was-
have 3 minutes and you paid for it. You guys want an encore? Boom, 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 boom. Right?
Do we need to worry about the S checkpoint? Because I know we said that there was 4
Yes.
and one was S, but then we never really talked
Oh,
about
no, nope. Yeah, so what we showed is that there's a famous checkpoint within S.
Mhm.
And it's a S-phase checkpoint to confirm that you're getting the job done.
Okay.
And we didn't say anything about it.
Okay.
Our book doesn't say anything about it either, I don't think. So, there are, there's more knowledge,
Mhm.
and we did a good job, introducing it. But yeah, nothing about the so-called S-phase checkpoint. And that's in S-phase toward the end of it, where you're like, let's get this wrapped up, folks. Yep, let's get all the final DNA copied and DNA repaired that might have made a mistake when you were copying it during S-phase. Yep. Yep, so now we should stop, and then we'll meet for our exam on Thursday. And, I've been asking questions outside of class, and if you don't have to hurry off to your next class, we can, go out in the hall and answer more questions. Yeah, because these are, all student questions are always great. Sometimes the instructor says, that's a good question. But that's, they should just, it's true, every question is that good. Yep. All right, cool. Yeah, how about that though? If your instructor is just up here texting and you're like, yeah, I want to talk about,
Just wanted to correct one thing.
yes, I'll just be texting while you talk.
So I have in my notes that P53 in